So unfortunately, I'm starting off with some disappointing news tonight that I'm not going to be talking about cookies, as the title may suggest. And that's because I have a different matter to discuss instead. My name is Monica. I am a neuroscientist as well, but I'm going to talk to you guys about poop. So we already know that fecal matter is loaded with bacteria, around 100 billion of these in a single gram of stool, which is about 300 times the US population. And these bacteria come from inside of your gut, where they normally live. And you and I both have our own unique population of bacteria inside of our intestines. And so, therefore, poop provides a snapshot of what's going on inside of our gut. And so, how does this exactly fit in with neuroscience? Well, if you ever had butterflies in your stomach, like I'm having right now, or if you've had trouble going to the bathroom when you're stressed about a deadline, you've met something called your gut-brain axis. And the gut-brain axis describes a chatty relationship, in which the brain can tell the gut when to digest, and the gut can tell the brain when it's hurting or when something else is wrong, which can lead us to being stressed or anxious. But don't forget about our bowel buddies, the gut-residing microbes. In fact, the little world inside of our gut, aka the gut microbiome, has a lot to say in this relationship, rightfully earning its place in the title, now referred to as the microbiota gut-brain axis. And so, why is this called a gut microbiome? Well, if you think back to 6th grade when we learned what a biome, like a desert or a rainforest biome is, it's an environment full of living and non-living things. Similarly, the gut microbiome is made up of living things, like the microbes themselves, and non-living things like their genes and their byproducts. That is different formatting, but that's okay. And so the reason why we care so much about their genes and their byproducts is because they have a strong influence on what your gut looks like. And to find out why, let's think of each microbe as a factory. Each gene would be an instruction manual in order to create a different product in this factory. And each product can do different things, like grab nutrients, break down food, and most importantly, send messages to other parts of the body. And your cells also have this similar machinery, so why do we care so much about the microbial ones? Well, for every human cell in your body, you have about 1.3 microscopic ones, and they outnumber our own by about 8 trillion. But what's more impressive than that number is that for every human gene, there are about 10 microbial genes, 10 times as many instruction manuals, belonging to microbes than us, because each type of microbe has a different library of manuals. And so your gut microbes matter. It's not just about your digestive system, even though they have a lot of influence on your diet and your energy use, but since your body is so interconnected, your gut microbes have influence on things like heart disease, immune health, and mental health. And because I am a neuroscientist who also has anxiety, I'm focusing on mental health today. And so one of the main components of our mental health is stress. If you are worried about something, you might feel sick to your stomach, or if you have a gut feeling that you've maybe forgot something, then you'll search your brain for what that is, and drive some more psychological trauma or stress. But what about more serious types of stress? What about trauma? More specifically, what about post-traumatic stress disorder, or PTSD? This is an anxiety disorder associated with serious traumatic events and characterized by symptoms like reliving trauma in nightmares and survivor's guilt. And when we think of these symptoms and we think of PTSD, a lot of us might think of soldiers or civilians in areas of conflict, like Ukraine and Palestine. Oh, okay. Unfortunately, the common thread between these populations is proximity to explosives. And so when people are near explosives, they experience something called a blast injury, which is characterized by poor memory, trouble moving, and pain, in addition to symptoms of PTSD. And these symptoms were all actually thought to be side effects of PTSD until blast injury was proven to be a traumatic brain injury in the 1990s. And the reason why it took until much later to figure out that it was actually a traumatic brain injury is because blast injury, specifically primary blast injury, is largely invisible. Instead of being hit with shrapnel or being burned by fire from an explosion, primary blast injury is like being hit with a pressure wave. And so I'm not a physicist, but essentially an explosion forces air around it to rapidly squish together and then rapidly expand again, to create a wave rippling around the epicenter. And this wave moves faster than the speed of sound. And so if this supersonic wave passes through a person, air and water-filled organs like your brain also get squished. And as you can imagine, your brain doesn't like that. And its natural defenses freak out and overreact, causing inflammation. But your brain isn't the only thing affected by blast injury. It is a full body injury. And despite most research focusing on the noggin, your brain isn't the only water-filled organ in your body. What about the lungs? What about air or water-filled organ, like the lungs or the stomach or the gut? And so which one would you think plays a bigger role in blast injury? Well, to answer this question, there was an experiment done about 15 years ago in which mice were separated into 3 groups before being exposed to a blast wave. One group had their brains shielded from this wave. Another had their body and protecting their internal organs from the wave. And the last group had no shielding at all. And as you can imagine, this last group, the one with no shielding, it had inflammation pretty much everywhere. And surprisingly, the one where the body was shielded only had no inflammation, but the one where we protected the brain looked just like the one with no shielding. And so the only group with any ability to prevent any inflammation was when we protected the body. And so the gut and other internal organs might have more influence on blast injury than it may get credit for. And we see this in veterans too, as some report gastrointestinal disorders after blast exposure, in addition to other symptoms we discussed before. And so there are many different sides of blast injury that kind of go hand in hand, that kind of survey across the body. But what about the organisms that live inside of our bodies? What about the gut microbiome? And so is the gut microbiome even affected by blast at all? And so to answer this, my lab took feces from mice with and without blast injury and identified different types of them and how many of each there are. This is actually the first time that the gut microbiome was characterized in this model of blast injury, and we see that there are definitely changes in microbial signatures from the blast. So yes, blast injury can traumatize our gut microbiome. But does this have anything to do with other symptoms of blast, like PTSD? And how do we even test PTSD in mice? Well, since PTSD is a type of anxiety, we will use one of the most common tests that we have for anxious behavior in rodents. This is called the open field test, where we put a mouse in a large open field, imagine like a kiddie pool, and see how much time they spend in different areas. So if we took a bird's eye view of said kiddie pool, we noticed that normal mice will sort of traverse around the area and aren't afraid to step into the center. However, blast mice or more anxious mice will want to hang out sort of around the edges. This is analogous to how someone like me with stage fright would much rather not be up here under the spotlight in front of a lot of people, and much rather be off to the side, sort of like by a wall or in a corner. And just like I'm being videoed right now, using video tracking with our mice, we can see how many things about their behavior, like their speed, their position, their stance, their location in this field, can give us an idea of how anxious these mice are, like their time in this outer ring. And I want to include a caveat that we understand that this is just one part of a very complex disorder like PTSD. And so to avoid getting too far ahead of ourselves, we try to use toned down terms like PTSD-like behaviors. But what's really cool is that in these mice with blast injury, these PTSD-like behaviors actually do correlate with changes in the gut microbiome. And I hope you caught onto that word correlate because it's very important, as we know that correlation is not causation. And looking at this graph, or for example, looking at this graph, you might be tempted to think that Paula Dane's influence on the American diet made the Washington, D.C. just a little bit richer, but logic would hopefully tell you that this is just a coincidence. Likewise, just because blast changed the gut microbiome and also elevated PTSD does not mean that the microbes themselves cause PTSD. They can just be happening together in parallel. And so how do we tell the difference? How can we tell if the gut microbiome is causing PTSD after blast or not? And so buckle up, because I'm going to share the most advanced, cutting-edge, science fiction-like technique that would have all the neuroscientists of the modern world begging UW to just hand me my doctorate already, because to prove causation, we have our mice, and we let them eat shit. And I say let them not because I'm making a habit of replacing pastries in famous sayings with fecal matter, but because mice do actually eat their own feces in a behavior known as coprophagy. And taking advantage of this evolutionary trait or effect, we can use poop to transplant microbiomes in a process known as fecal microbiota transplant, or FMT. And so to give a mouse a new microbiome, we first have to get rid of the one it already has with drinkable antibiotics. And then we feed it a new one. Traditionally, this is done by mixing feces with salt water and pushing it down a mouse's esophagus while it's being restrained. And as you can imagine, this is highly stressful for the animal, which is not only bad for animal welfare, but also it makes studying stress a bit complicated. And so to avoid these problems, I'm developing a stress-free alternative, introducing the poopsicle. It is exactly what it sounds like. It is a frozen fecal treat made up of nutritional jello, happy microbe juice, and the most important ingredient of all, poop. And so compared to the syringe method, mice can voluntarily eat this poopsicle, which reduces the stress of being restrained. As you can see, this started off very much as a joke, but the professor that I work with likes it so much that it started making its way into the official paperwork. So I guess we're sticking to the bit. And so back to our instructions. We can now take poop from a donor mouse, make it into a poopsicle that the recipients who received the antibiotics before can now eat. And now, since we're interested in blast-induced changes to the gut microbiome, the donor mice that we're going to use are going to be ones with blast injury. And so we're back to our original question, causation. Can we transfer PTSD via poopsicle? In other words, is the blast-exposed gut microbiome responsible for driving PTSD symptoms? kind of, maybe, sort of. Initial tests show that if we transfer poop from a mouse with blast injury to a naive mouse via poopsicle, the recipient will show increased PTSD-like behaviors. However, there's a lot more work that needs to be done to validate this claim, a lot more power, if you will. And so, now what? Clearly, the goal of my project is not to give PTSD to mice via poop, that it's not helpful for anyone or any animal, but rather I'm going to try to do the opposite. I want to give mice exposed to blast that now have PTSD a poopsicle from a healthy donor and see if it improves their recovery. And this will give us an idea of if and how targeting the gut microbiome leads to therapeutic results that we can apply to humans. Now you might be thinking that feeding poop will never be a real thing for people to do, but for better or worse, you would be wrong. The first record of fecal microbiota transplants being used in people was from 1700 years ago in ancient China to cure diarrhea. Nowadays, another diarrhea-inducing bacterial infection known as C. difficile is usually treated with antibiotics, which can fail about 20 to 60% of the time. And so for these cases of antibiotic-resistant infection, they can get new feces via colonoscopy through this FMT method. And this cures infection with over a 90% success rate. And of course, this is one application of FMTs with behavioral or neuropsychiatric disorders like PTSD. It's a little bit more complicated. And also there are limitations of animal research, but especially in questions involving multiple full-body systems like blast injury, animals are needed to understand these complicated relationships. And findings from animal studies translate more to human research than you might think. For example, just as mice do not like stressful procedures like force feeding, some people may feel squeamish at the idea of some clinical procedures of FMT, like the colonoscopies I mentioned earlier, or something called a nasoduodenal tube. Not fun. And so just like I've developed an alternative method of the poopsicle for mice, scientists are also working on creating something like poop pills or capsules to offer a less invasive alternative for people. And if poop is just not your thing, there are also non-poop related therapies being worked on to try to see if diet-based therapies or treatments can influence the gut microbiome and thus improve symptoms, which, if proven true, can offer much more accessible treatments or treatment routes for people with PTSD or other mental health struggles. And so to recap, if you give a mouse a new microbiome, poopsicle, you might find that although the gut is an unlikely place to target mental health-focused therapies, it could just be that the proof is in the poop. Thank you.